10. Porphyrins and the basis of life. I see little hope to be able to explain the subtle difference between a normal and a sick cell as long as we do not understand the basic difference between a cat and a stone. Albert Szent-Györgyi. Strangely enough, porphyrin is not a household word. It is not a sugar, fat, or protein, nor is it a vitamin, mineral, or hormone. But it is more basic to life than any other of life's components, because without it, we would not be able to breathe. Plants could not grow. There would not be any oxygen in the atmosphere. Wherever energy is transformed, wherever electrons flow, they look for porphyrins. When electricity alters nerve conduction or interferes with the metabolism of our cells, porphyrins are centrally involved. As I write this chapter, a dear friend has just died. For the last 7 years, she had to live without electricity, hardly ever seeing the sun. She seldom ventured out in the daytime. When she did, she covered herself from head to foot in thick leather clothing, a broad-brimmed leather hat hiding her face, and glasses bearing 2 layers of dark lenses concealing her eyes. A former dancer who loved music, nature, and the outdoors, Bethany was virtually abandoned by a world in which she no longer belonged. Her condition, probably caused by her years of work for a computer company, was a classic example of an illness that has been known to medicine only since 1891. Its emergence at that time being one of the side effects of the sudden worldwide expansion of electrical technology. Its connection with electricity was discovered a century later, although it is now considered an extremely rare genetic disease affecting as few as 1 person in 50,000. Porphyria was originally thought to affect as many as 10% of the population. Its supposed rarity is due in large part to the ostrich-like behavior of the medical profession after World War II. In the late 1940s, medical practitioners were staring at an impossible contradiction. Most synthetic chemicals were known poisons, but one of the legacies of the war was the ability to manufacture products from petroleum easily and cheaply to substitute for almost every consumer product imaginable. Now, thanks to the fledgling petrochemical industry bringing us better living through chemistry, synthetic chemicals were going to be literally everywhere. We were going to be wearing them, sleeping on them, washing our clothes, our hair, our dishes, and our homes with them, bathing in them, insulating our houses with them, carpeting our floors with them, spraying our crops, our lawns, and our pets with them, preserving our food with them, coating our cookware with them, packing our groceries in them, moisturizing our skin with them, and perfuming our bodies with them. The medical profession had 2 choices. It could have attempted to study the health effects singly and in combination of the hundreds of thousands of new chemicals that were kaleidoscoping over our world, a virtually impossible task. The attempt itself would have put the profession on a collision course with the mushrooming petrochemical industry, threatening the banning of most new chemicals and the strangling of the economic boom of the next 2 decades. The other alternative was for the profession to bury its collective head in the sand and pretend that the world's population was not actually going to become poisoned. Environmental medicine was born as a medical specialty in 1951, founded by Dr. Theron Randolph. It had to be created. The scale of the poisoning was too great to go completely ignored. The sheer numbers of sickened patients abandoned by mainstream medicine produced an urgent need for practitioners trained to recognize at least some of the effects of the new chemicals and to treat their resulting diseases. But the specialty was ignored by the mainstream as though it didn't exist. Its practitioners ostracized by the American Medical Association. When I attended medical school from 1978 to 1982, environmental medicine wasn't even on the curriculum. Chemical sensitivity, the unfortunate name that has been given to the millions of poisoned patients, was never mentioned at school. Neither was porphyria, arguably a more appropriate name. It still isn't mentioned, not in any medical school in the United States. Heightened sensitivity to chemicals, we recall, was first described by New York physician George Miller Baird, who considered it a symptom of a new disease. The initial electrification of society through telegraph wires brought with it the constellation of health complaints known as neurasthenia, two of which were a tendency to develop allergies and a drastically reduced tolerance for alcohol and drugs. By the late 1880s, insomnia, another prominent symptom of neurasthenia, had become so rampant in Western civilization that the sale of sleeping pills and potions became big business, with new formulations coming on the market almost every year. Bromides, paraldehyde, chloral amylhydrate, urethane, hypnol, somnol, cannabinol, and other hypnotics flew off pharmacists' shelves to satisfy the frustrated urge to sleep and the addiction that so often allowed the long-term use of these drugs. In 1888, one more drug was added to the list. Sulfonal was a sleeping medication that had a reputation for its prompt effect, its non-addictive nature, and its relative lack of side effects. There was just one problem, which only became widely known after 3 years of its popularity. It killed people. But its effects were quirky, unexpected. 9 people could take sulfonal, even in large doses and for a long time with no untoward effects. But the 10th person, sometimes after only a few or even one small dose, would become critically ill. He or she would typically be confused, so weak as to be unable to walk, constipated, with pain in the abdomen, sometimes with a skin rash, and reddish urine often described as the color of port wine. Their reactions were idiosyncratic, liable to affect almost any organ, and the patients were apt to die of heart failure without warning. Between 4 and 20% of the general population were reported to be subject to such side effects from taking sulfonal. During the ensuing decades, the chemistry of this surprising disease was worked out. Porphyrins are light, sensitive pigments that play pivotal roles in the economy of both plants and animals, and in the ecology of planet Earth. In plants, a porphyrin bound to magnesium is the pigment called chlorophyll, that makes plants green and is responsible for photosynthesis. In animals, an almost identical molecule bound to iron is the pigment called heme, the essential part of hemoglobin that makes blood red and enables it to carry oxygen. It is also the essential part of myoglobin, the protein that makes muscles red and delivers oxygen from our blood to our muscle cells. Heme is also the essential component of cytochrome C and cytochrome oxidase enzymes that are contained in every cell of every plant, animal, and bacterium that transport electrons from nutrients to oxygen so that our cells can extract energy. And heme is the main component of the cytochrome P450 enzymes in our liver that detoxify environmental chemicals for us by oxidizing them. In other words, porphyrins are the very special molecules that interface between oxygen and life. They are responsible for the creation, maintenance, and recycling of all of the oxygen in our atmosphere. They make possible the release of oxygen from carbon dioxide by plants, the extraction of oxygen back out of the air by both plants and animals, and the use of that oxygen by living things to burn carbohydrates, fats, and proteins for energy. The high reactivity of these molecules, which makes them transformers of energy, and their affinity for heavy metals, also makes them toxic when they accumulate in excess in the body, as happens in the disease called porphyria, a disease that is not really a disease at all, but a genetic trait, an inborn sensitivity to environmental pollution. Our cells manufacture heme from a series of other porphyrins, and porphyrin precursors in a series of 8 steps catalyzed by 8 different enzymes. Like workers on an assembly line, each enzyme has to work at the same rate as all the others in order to keep up with the demand for the final product, heme. A slowdown by any one enzyme creates a bottleneck, and the porphyrins and precursors that accumulate behind the bottleneck get deposited all over the body, causing disease. Or if the first enzyme is working harder than the rest, it produces precursors faster than the enzymes down the line can handle, with the same result. The accumulation in the skin can cause mild to disfiguring skin lesions and mild to severe light sensitivity. The accumulation in the nervous system causes neurological illness, and the accumulation in other organs causes corresponding illness. And when excess porphyrins spill into the urine, it takes on the color of port wine. Because porphyria is assumed to be so rare, it is almost always misdiagnosed as some other disease. It is fairly called the little imitator because it can affect so many organs and mimic so many other conditions. Since patients usually feel so much sicker than they look, they are sometimes wrongly through to have psychiatric disorders and to often wind up on mental wards. And since most people don't carefully examine their own urine, they usually fail to notice its reddish hue, particularly since the color may be evident only during severe disabling attacks. The enzymes of the heme pathway are among the most sensitive elements of the body to environmental toxins. Porphyria, therefore, is a response to environmental pollution and was indeed extremely rare in an unpolluted world, except for one severe, disfiguring congenital form, of which only a few hundred cases are known in the world. Porphyrin enzyme deficiencies do not normally cause disease at all. Human beings are genetically diverse, and in times past, most people with relatively lower levels of one or more porphyrin enzymes were simply more sensitive to the environment. In an unpolluted world, this was a survival advantage, allowing the possessors of this trait to easily avoid places and things that might do them harm. But in a world in which toxic chemicals are inescapable, the porphyrin pathway is to some degree always stressed, and only those with high enough enzyme levels tolerate the pollution well. Sensitivity has become a curse. Because of the way it was discovered and the lack of synthetic chemicals in the environment at that time, porphyria became known as a rare disease that was triggered in genetically susceptible people by certain drugs such as sulfonal and barbiturates, which these patients had to avoid. It was not until another century had passed in the early 1990s that Dr. William E. Morton, professor of occupational and environmental medicine at Oregon Health Science University, realized that because ordinary synthetic chemicals were far more widespread in the modern environment than pharmaceuticals, they had to be the most common triggers of porphyric attacks. Morton proposed that the controversial disease called multiply chemical sensitivity (MCS) was in most cases identical with one or more forms of porphyria. And when he began testing his MCS patients, he found that, indeed, 90% of them were deficient in one or more porphyrin enzymes. He then investigated a number of their family trees, looking for the same trait, and succeeded in demonstrating a genetic basis for MCS, something no one had attempted before because MCS had never before been connected to a testable biological marker. Morton also found that most people with electrical sensitivity had porphyrin enzyme deficiencies, and that electrical and chemical sensitivities appear to be manifestations of the same disease. Porphyria, Morton showed, is not the extremely rare illness it is currently thought to be, but has to affect at least 5 to 10% of the world's population. Morton was courageous because the rare disease world of porphyria had come to be dominated by a handful of clinicians who controlled virtually all research and scholarship in the small inbred field. They tended to diagnose porphyria only during acute attacks with severe neurological symptoms and to exclude cases of milder, smoldering illness. They generally would not make the diagnosis unless porphyrin excretion in urine or stool was at least 5 to 10 times normal. This makes no sense, wrote Morton in 1995, and would be analogous to this restricting the diagnosis of diabetes mellitus to those who have ketoacidosis, or restraining the diagnosis of coronary artery disease to those who have myocardial infarction. The high numbers reported by Morton agree with the numbers reported over a century ago. The proportion of the population that became ill when they took the sleeping medication sulfonal. They are consistent with the finding in the 1960s of movefactor, a lavering staining chemical not only in the urine of patients diagnosed with porphyria, but in the urine of 5 to 10% of the general population. Movefactor was eventually identified as a breakdown product of porphobilinogen, one of the porphyrin precursors. Morton also found, in agreement with recent reports from England, the Netherlands, Germany, and Russia, that persistent neurological problems occur during the chronic, smoldering phase of every type of porphyria, even those types which were previously supposed to cause only skin lesions. Hans Günther, the German doctor who in 1911 gave porphyria its name, stated that such individuals are neuropathic and suffer from insomnia and nervous irritability. Morton has brought us back to the original view of porphyria. It is not only a fairly common disease, but exists most often in a chronic form with comparatively mild symptoms. And its principal cause is the synthetic chemicals and electromagnetic fields that pollute our modern environment. Porphyrins are central to our story not only because of a disease named porphyria, which affects a few percent of the population, but because of the part porphyrins play in the modern epidemics of heart disease, cancer, and diabetes, which affect half the world. And because their very existence is a reminder of the role of electricity in life itself, a role which a few courageous scientists have slowly elucidated. As a child, Albert St. Georgi, pronounced approximately like St. Georgie, hated books and needed a tutor's help to pass his exams. But later, having graduated from Budapest Medical School in 1917, he went on to become one of the world's greatest geniuses in the field of biochemistry. In 1929, he discovered vitamin C, and during the next few years, he worked out most of the steps in cellular respiration, a system now known as the Krebs cycle. For these two discoveries, he was awarded the Nobel Prize in Physiology or Medicine in 1937. He then spent the next 2 decades figuring out how muscles function. After emigrating to the United States and settling at Woods Hole, Massachusetts, he received the Albert Lasker Award of the American Heart Association in 1954 for his work on muscles. But perhaps his greatest insight is one for which he is least known, although he devoted almost half his life to the subject. For on March 12, 1941, in a lecture delivered in Budapest, he boldly stood up before his peers and suggested to them that the discipline of biochemistry was obsolete and should be brought into the 20th century. Living organisms, he told them, were not simply bags of water in which molecules floated like tiny billiard balls, forming chemical bonds with other billiard balls with which they happened to collide. Quantum theory, he said, had made such old ideas invalid. Biologists needed to study solid-state physics. In his own specialty, although he had worked out the structures of the molecules involved in muscular contraction, he could not begin to fathom why they had those particular structures, nor how the molecules communicated with one another to coordinate the activities. He saw such unsolved problems everywhere he looked in biology. One of my difficulties within protein chemistry, he bluntly told his colleagues, was that I could not imagine how such a protein molecule can live. Even the most involved protein structural formula looks stupid, if I may say so. The phenomena that had forced Szent-Györgyi to face these questions were the porphyrin-based systems of life. He pointed out that in plants, 2,500 chlorophyll molecules form a single functional unit, and that in dim light at least 1,000 chlorophyll molecules have to cooperate simultaneously in order to split one molecule of carbon dioxide and create one molecule of oxygen. He spoke about the enzymes of oxidation, the cytochromes in our cells, and wondered again how the prevailing model could be correct. How could a whole series of large protein molecules be arranged geometrically so that electrons could wander directly from one to the other in a precise sequence? Even if we could devise such an arrangement, he said, it would still be incomprehensible how the energy liberated by the passing of an electron from one substance to the other, viz. From one iron atom to the other, could do anything useful. Szent-Györgyi proposed that organisms are alive because thousands of molecules form single systems with shared energy levels such as physicists were describing in crystals. Electrons don't have to pass directly from one molecule to another, he said. Instead of being attached to only one or two atoms, electrons are mobile, belong to the whole system, and transmit energy and information over large distances. In other words, the stuff of life is not billiard balls, but liquid crystals and semiconductors. Szent-Györgyi's sin was not that he was incorrect. He wasn't. It was his failure to respect the old animosity. Electricity and life were long divorced. The Industrial Revolution had been running full bore for a century and a half. Millions of miles of electric wires clothed the earth, exhaling electric fields that permeated all living things. Thousands of radio stations blanket the very air with electromagnetic oscillations that one could not avoid. Skin and bones, nerves, and muscles were not allowed to be influenced by them. Proteins were not permitted to be semiconductors. The threat to industry, economics, and modern culture would be too great. So, biochemists continued to think of proteins, lipids, and DNA as though they were little marbles drifting in a watery solution and colliding with one another at random. They even thought of the nervous system this way. One forced to the admitted parts of quantum theory, but only on a limited basis. Biological molecules were still only permitted to interact with the immediate neighbors, not to act at a distance. It was okay to acknowledge modern physics only that much like opening a small hole in a dam for knowledge to leak through one drop at a time, while the main structure is reinforced lest a flood demolish it. All knowledge about chemical bonds and enzymes in a water solution must now coexist with new models of electron transport chains. It was necessary to invest these to explain phenomena that were almost central to life. photosynthesis, and respiration. Large porphyrin containing protein molecules no longer had to move and physically interact with one another in order for anything useful to happen. These molecules could stay put and electrons could shuttle between them instead. Biochemistry was becoming that much more life, but it still had a long way to go. For even in the new models, electrons were constrained to move only like little messenger boys between one protein molecule and its immediate neighbor. They could cross the street, so to speak, but they couldn't travel down a highway to a distant town. Organisms were still pictured essentially as bags of water containing very complex solutions of chemicals. The laws of chemistry had explained a lot about metabolic processes, and electron transport now explained even more. But there was not yet an organizing principle. Elephants grow from tiny embryos. We grow from single brainless cells. Salamanders regenerate perfect limbs. When we are cut or break a bone, cells and organs throughout our body mobilize and coordinate the activities to repair the damage. How does the information travel? How, borrowing St. George's words, do proteins live? Despite St. George's sin, his predictions have proven correct. Molecules in cells do not drift at random to collide with one another. Most are firmly anchored to membranes. The water inside cells is highly structured and does not resemble the free-flowing liquid that sloshes around in a glass before you drink it. Piezoelectricity, a property of crystals that makes them useful in electronic products, that transforms mechanical stress into electrical voltages and vice versa, has been found in cellulose, collagen, horn, bone, wool, wood, tendon, blood vessel walls, muscle, nerve, fibrin, DNA, and every type of protein examined. In other words, something most biologists have been denying for 2 centuries: electricity is essential to biology. St. George was not the first to challenge conventional thinking. It was Otto Lehmann already in 1908 who, noticing the close resemblance between the shapes of known liquid crystals and many biological structures, proposed that the very basis of life was the liquid crystalline state. Liquid crystals, like organisms, had the ability to grow from seeds, to heal wounds, to consume other substances or other crystals, to be poisoned, to form membranes, spheres, rods, filaments, and helical structures, to divide, to mate with other forms, resulting in offspring that had characteristics of both parents, to transform chemical energy into mechanical motion. After St. George's daring Budapest lecture, others pursued his ideas. In 1949, Dutch researcher E. Katz explained how electrons could move through a semiconducting chlorophyll crystal during photosynthesis. In 1955, James Bessam and Melvin Calvin, working for the U.S. Atomic Energy Commission, elaborated on this theory. In 1956, William Arnold at Oak Ridge National Laboratory confirmed experimentally that dried chloroplasts, the particles in green plants that contain chlorophyll, have many of the properties of semiconductors. In 1959, Daniel Alley at Nottingham University proved that dried proteins, amino acids, and porphyrins are indeed semiconductors. In 1962, Roderick Layton, also at Oak Ridge, found that photosynthetic tissues in living plants behave like semiconductors. In 1970, Alan Adler at the New England Institute showed that thin films of porphyrins do also. In the 1970s, chemist Freeman Cope at the United States Naval Air Development Center in Worcester, Pennsylvania, emphasized the importance of solid-state physics for a true understanding of biology, as did biologist Alan Frey, the most active American researcher into the effects of microwave radiation on the nervous system at the time. Ling Wei, professor of electrical engineering at the University of Waterloo in Ontario, stated boldly that a nerve axon is an electrical transmission line and that its membrane is an ionic transistor. He said that the equivalent circuitry can be found in any electronics book today, and that one can easily derive the nerve behavior from semiconductor physics. When he did so, his equations predicted some of the properties of nerves that were, and still are, puzzling to physiologists. In 1979, a young professor of bioelectronics at the University of Edinburgh published a book titled Dielectric and Electronic Properties of Biological Materials. The earlier work of Ali and Arnold had been criticized because the activation energies they had measured, the amount of energy necessary to make proteins conduct electricity, seemed to be too large. Supposedly, there was not enough energy available in living organisms to lift electrons into the conduction band. Proteins might be made to conduct electricity in the laboratory, said the critics, but this could not happen in the real world. Ali and Arnold, however, had done all their work on dried proteins, not living ones. The young professor, Ronald Pething, pointed out the obvious. Water is essential to life, and proteins become more conductive if you added water to them. In fact, studies had shown that adding only 7.5% water increased the conductivity of many proteins 10,000-fold or more. Water, he proposed, is an electron donor that dopes proteins and turns them into good semiconductors. The electronic role of living water had already been noted by others. Physiologist Gilbert Link, realizing that cell water is a gel and not a liquid, developed his theory of the electronic nature of cells in 1962. More recently, Gerald Pollack, professor of bioengineering at the University of Washington, had taken up this line of investigation. He was inspired by Ling when they met at a conference in the mid-1980s. Pollack's most recent book, The Fourth Phase of Water, Beyond Solid, Liquid, and Vapor, was published in 2011. The late geneticist Maya Wan Ho in London has clothed St. George's ideas in garments that all can see. She developed a technique using a polarizing microscope that displayed, in vivid color, the interference patterns generated by the liquid crystalline structures that make up living creatures. The first animal she put under her microscope was a tiny worm, a fruit fly larva. As it crawls along, it weaves its head from side to side, flashing jaw muscles in blue and orange stripes on a magenta background. She wrote in 1993 in her book, The Rainbow and the Worm, The Physics of Organisms. She and many others have urged that the liquid crystalline properties of our cells and tissues not only teach us about our chemistry, but have something special to tell us about life itself. Vladimir Sedlak, pursuing St. George's ideas in Poland, developed the discipline of bioelectronics within the Catholic University of Lublin during the 1960s. Life, he said, is not only a collection of organic compounds undergoing chemical reactions, but those chemical reactions are coordinated with electronic processes that take place in an environment of protein semiconductors. Other scientists working at the same university are continuing to develop this discipline theoretically and experimentally today. Marian Vnuk has focused on porphyrins as key to the evolution of life. He states that the principal function of porphyrin systems is an electronic one. Joseph Zahn, head of the Department of Theoretical Biology at the university, has focused on the electronic properties of biological membranes. Oddly enough, the use of porphyrins in electronic products instructs us about biology. Adding thin films of porphyrins to commercially available photovoltaic cells increases the voltage, current, and total power output. Prototype solar cells based on porphyrins have been produced, as have organic transistors based on porphyrins. The properties that make porphyrins suitable in electronics are the same properties that make us alive. As everyone knows, playing with fire is dangerous. Oxidation releases tremendous energy quickly and violently. How, then, do living organisms make use of oxygen? How do we manage to breathe and metabolize our food without being destroyed in a conflagration? The secret lies in the highly pigmented fluorescent molecule called porphyrin. Strong pigments are always efficient energy absorbers, and if they are also fluorescent, they are also good energy transmitters. As St. George taught us in his 1957 book Bioenergetics, fluorescence thus tells us that the molecule is capable of accepting energy and does not dissipate it. These are two qualities any molecule must have to be able to act as an energy transmitter. Porphyrins are more efficient energy transmitters than any other of life's components. In technical terms, their ionization potential is low and their electron affinity high. They are therefore capable of transmitting large amounts of energy rapidly in small steps, one low energy electron at a time. They can even transmit energy electronically from oxygen to other molecules, instead of dissipating that energy as heat and burning up. That's why breathing is possible. On the other side of the great cycle of life, porphyrins in plants absorb the energy of sunlight and transport electrons that change carbon dioxide and water into carbohydrates and oxygen.